All right, so hi everyone, and welcome to our first webinar of Immunoassay Applications Week. So this week we will be hosting live workshops every day, and each workshop will focus on tips and best practices for a specific immunoassay. So today's focus is, of course, going to be flow cytometry, and I'm really excited to have Dr. Alastair McKee with us, and he's going to be leading today's workshop. But before I go ahead and pass it off to Ali, I just want to do a quick introduction of Proteintech and share some of our latest innovations. So Proteintech is known as a leading provider of the life science reagents. So the company was founded back in 2001, and at that time it was a primary supplier of antibodies. And so because we have been in the game for a really long time, we have about 2/3 of the human proteome covered. So we have antibodies for against over 13,000 targets. We also offer immunoassays such as ELISA kits and IHC kits, as well as cell separation kits and antibody labeling kits. The company then later expanded to include more product lines, so we also have our humankind branded cytokines and growth factors. So all our proteins are actually going to be expressed in human HEC293 cells so that you can be confident that they have the native folding, post-translational modifications, and glycosylation that you would expect to see in human proteins, and that ultimately increases their bioactivity compared to proteins that are, for example, raised in E. coli systems. We also have our Chromatech branded nanobody based reagents. So, nanobodies are about 10 times smaller than traditional antibodies, and they can bind to their targets with really high affinity, making them ideal for certain applications such as immunoprecipitation, immunofluorescence, especially if you're doing something like super resolution microscopy, and protein purification. And then lastly, we also have a subsidiary of Proteintech known as Proteintech Genomics, and they manufacture antibodies that are conjugated to oligonucleotides for single cell RNA sequencing. So, as a company that is driven by scientists, we're here to support our researchers and scientific community any way that we can. So, our award winning products are self manufactured, and we offer the largest in house portfolio of antibodies for the human proteome. For convenience and testing, most of our antibodies are also offered in 20 microliter trial sizes. We also know the importance of product validation, and we offer open access validation data. So, whenever we are testing new products, we validate with relevant sample types, appropriate experimental conditions, and using knockout knockdown models whenever possible. Also, as antibody experts, we have in depth knowledge of immunoassay applications, and we can offer outstanding technical support for our customers. We also stay on top with cutting edge technology. So, for example, we have our proprietary antigen specific B cell cloning and engineering platform, which we use to manufacture our recombinant antibodies. Also, with 7 inventory sites and manufacturing and research sites on 3 continents, we are able to offer next day delivery to many areas in the world. So, as a company, we do measure our success by our customers' scientific success, specifically by helping them publish impactful research faster. So, this graph here on the left shows that over our company history, our products are featuring in more and more publications each year. And as of now, we have 350,000 publication citations, making us one of the top 5 cited antibody companies and also one of the fastest growing companies in product citations. I also want to highlight that as a company, we strive to make the searchability of our products on our website as user friendly and informative as possible. So, earlier this year, we actually launched some new updates on our website to help our customers select the right antibody. So, this includes our unique 3D epitope viewer, which allows you to interact with epitope structures and explore antibody binding sites so you know exactly where our antibodies bind to your protein of interest. We've also introduced advanced filter options to help you find exactly what you need. For example, we have our immunogen region selector toggle, which allows you to filter for antibodies that are generated from your region of interest. And now you also no longer have to filter through all the variations of an antibody from the search results. You can view all formats of an antibody, including things like conjugations and buffer formulations, on just one page. And the idea for this was to help streamline your process for antibody selection. And then lastly, I just want to highlight real quick that we've also built off the information from our epitope mapper and database, and we've recently launched an AI-powered search tool on our website called Able. So Able allows you to directly chat and ask for product recommendations, help for experimental design, and access relevant validation data, citations, and resources. So for example, you could ask Able, what CD4 antibody can I use for flow cytometry in human PBMCs? And Able will suggest the best product for your experimental needs so that you don't have to spend countless hours searching for the right antibody. So this AI-powered tool is actually the first of its kind, and it was designed by our scientists in-house with our customers in mind so that you all can focus on your discoveries and not on reagent selection. So if you do have some time to check it out, you can access it by scanning this QR code here, or you can just simply go onto our website at ptglab.com, and Able lives right next to the search box. So with that, I would like to introduce our speaker for today's talk. So Alastair obtained his PhD at the Quadram Institute in Norwich, UK, where he investigated the effects of an altered gut microbiota on the progression of breast cancer in a preclinical animal model. Following his studies, he worked as a flow cytometry scientist for 2 years at an antibody manufacturing company, where he was responsible for putting products through their paces to ensure high target specificity, reproducibility, and overall product quality before he joined us as an account manager for Proteintech. Alistair's passion for flow cytometry stems from the depth, diversity, quantity, and quantifiable nature of the data this application allows scientists to collect. His thoughts are that it's an ever-evolving application which is continuously offering researchers more and more in terms of the questions they can ask and answer by combining the basic principles of both microfluidics and fluorescence detection. So with that, I will go ahead and pass it off to you, Ali.
Thank you very much, Afrida. Let me see if I can get this shared. And hopefully this should go into full screen mode in a sec. Okay, I'm hoping you can see that in full screen mode. Yeah, big thank you for that introduction, Frida. Yes, flow cytometry really is one of my favorite immunoassay applications during my studies and also my time even in industry. I used several immunoassays, and flow cytometry is one that stood out to me just because of the kind of data that you can generate and the various uses that you can put flow to. So just for a quick overview of what to expect from the talk this evening, if you're in the UK or this morning if you're in the US and anywhere else in the world, welcome as well. So we're going to start with a quick introduction to flow cytometry, what it is and what you might use it for. I'd like to do a little bit about how flow cytometers work. I think they're pretty phenomenal bits of kit, so it's useful just to get an idea of the setup and what your samples are going through. Then we're going to spend most of our time looking at experimental planning and protocol considerations before we move on to controls, which within flow cytometry are absolutely crucial. And then we're going to have some time to go through some troubleshooting and, of course, have a Q&A towards the end. So with that, I'm going to jump into an introduction. So what is flow cytometry? Essentially, you are passing a suspension of single cells, which you've labeled with fluorescent probes. Usually those would be antibodies conjugated to fluorophores. You're passing these through a microfluidic system, which is associated with fluorescence-based optical detectors. So these detectors are detecting the presence of those probes on single cells, and single cells really is the key, because this is what allows us to look at one cell at a time within usually a heterogeneous population of cells. What can you use it for? Predominantly, flow cytometry is used in immunophenotyping. So this would be the process of trying to identify different cell types within a heterogeneous sample mix, so something containing multiple different cells. Usually, obviously, using antibodies to do that, but you can use flow cytometry in an array of different applications. So cell cycle analysis, for example, looking at apoptosis, proliferation, mitosis. You can, of course, measure relative protein expression, so just looking at one particular target and seeing how the expression of that protein changes relative to maybe a control sample. And of course, you can use flow cytometry for cell isolation as well. But what does a flow cytometry experiment actually involve? So when I think about setting up an experiment, I kind of break it down into these 4 different parts. So the first part is experimental planning. And without doubt, this is the area that I would hope that you and I certainly did spend most of my time thinking about when it came to my experiments, particularly if you're working with larger antibody panels. Sample preparation and immunostaining is obviously the day in the lab at the bench doing your sample prep and staining your samples with your antibody panel. And then downstream, you've got the data collection on your cytometer and analysis as well to get the crucial data that you're looking for. But for the purpose of today, we're going to focus on these kind of these first two steps to this kind of process. Okay, but before we dive into that, let's just learn a little bit about how flow cytometers work. So I want to first start with kind of introducing the microfluidic system within a flow cytometer. So it's really crucial that when we are running our samples that we pass cells through the cytometer, past the lasers or past the interrogation point one at a time. And in order to get them past the lasers one at a time, we need this microfluidic system to form a single file stream of cells down what we would call the core stream. So on the right-hand side, you've got a kind of depiction of what is sort of happening within your cytometer. And at the top, you can see we're injecting our sample containing many different cell types into the cytometer, and it's forming this core sample flow. And on either side of that, we have something called sheath fluid. And sheath fluid basically encapsulates our sample core to make sure that our cells are in single file. How does it do that? Well, effectively, it's called hydrodynamic focusing. So on the bottom left, I've got a very simple graphic which kind of depicts how you might imagine to see the flow of water in a river. And if you stood next to a river and you threw a stick maybe into the middle of the river and you dropped a stick at the banks of the river on the side, what you'd find is that the stick in the middle of the river is likely to flow a lot faster than the stick at the edge. And the reason for that is because of the hydrodynamic forces being applied to the water molecules as the water is flowing down the river. So effectively, the banks slow down that flow of water. And in the context of flow cytometry, that can be a problem, because what it would do is prevent or it would encourage cells flowing in the middle to flow faster and the cells on the sides to flow slower, which might ultimately end up in more than one cell passing your laser or your interrogation point at a given time, which is what we want to avoid. And what sheath fluid does is it kind of takes on those hydrodynamic forces and allows the sample core to flow at the same rate, allowing cells to pass their detectors all at the same time, pass their lasers all at the same time. In terms of our optics and our detection, so each flow cytometer is going to be equipped with several different lasers. This is obviously hardware dependent, so depending on the flow cytometer you're using, you will have fewer or more lasers of different wavelengths. But the job of these lasers really is to excite the fluorophores that you label your cells with, causing them to absorb the energy and emit photons of a known kind of wavelength of light. And those photons are going to be directed through your detection system by what we call dichroic filters, which only allow light of specific wavelengths through onto their detectors. And when those photons or that light strikes the detector, it's going to generate voltage signal, so like an electrical signal. And effectively, the more photons or light that you are causing emission of from a particular cell, the higher the voltage signal you'll have. And you can get that as a measure then to get an idea of how much protein is present in or on your cell. But I just want to kind of give a bit more detail as to what exactly is happening as your cell is passing through a laser. So this is happening obviously for each laser, but I think when we think about flow, because things happen so quickly through this microfluidic system, we kind of think that everything happens in an instant. And the reality is it doesn't. The cells pass through the laser over a period of time. And as it passes through that laser, there's going to be a change in the amount of voltage that we pick up our detectors over that time. And we can kind of plot that as a histogram. So in this example here, we've got a cell entering our laser or our interrogation point. And as it does so, it starts to either emit photons or scatter light photons, which generates this voltage. And as time goes on, that cell enters the main part of the laser, generating increased amount of voltage as more of the cell is exposed to the laser. And slowly, as it passes through the interrogation point, that voltage will drop. So it creates this nice voltage over time. And that is effectively what we're plotting for each interaction between a cell and our interrogation point, and for each fluorophore as well. So what kind of data are we collecting? There's a host of different data points that you collect when you're running a cytometry experiment, but the two major points really are light scatter and fluorescence. So light scatter is going to give us some morphological information about a given event or a cell in terms of the cell size and the granularity of that cell, i.e. what are the kind of components like within that cell. And then fluorescence, of course, is the information we're getting on the relative expression of a particular protein, and we get that from our immunostaining, so using antibodies conjugated to fluorophores to stain those particular proteins. In terms of the difference between forward and side scatter, just to kind of depict exactly what's happening. So again, kind of visiting the idea of a cell entering a laser. So forward scatter is going to give us an idea of size, and effectively how it measures that is the larger the cell passing through an interrogation point or passing through a laser, the more light is going to be scattered behind that kind of cell. So as the larger cell passes through, light is scattered over it. That's picked up by a forward scatter detector, and the larger that cell is, the more light is scattered. So you get an increased voltage over time when you're plotting that particular cell. So just an example here with a large cell passing through, there's an increased amount of scattered light striking the detector compared to a smaller cell where there's less scattered light striking the detector. And side scatter, as I say, is kind of offering information about the granularity of your cells, so the different organelles, the granules, so things like vesicles and that kind of thing. The more of those present in your cell, the more side scatter you're going to see. So again, very similarly to forward scatter, your light is shining onto, or your laser is shining onto your cell. As it interacts with that cell, it starts to scatter it not just forward, but in many, many different directions. And your side scatter detector is going to pick up that scatter that's not directly behind the cell. And again, the more granular your cell is, so the more bits that you have inside the cell, the larger that voltage over time is going to be. And again, that's going to be data that we can plot downstream. So just as a kind of very basic idea of exactly what you'd be looking at in terms of what forward and side scatter can offer you. On the left here, we've got an example of PBMC cells, so cells from blood, which contain lymphocytes and monocytes. Monocytes are known to be slightly larger cells. They're also more granular, so naturally they'll sit further along this forward side scatter axis. Maybe I should turn on my pointer very quickly. So they'll sit slightly further along the forward scatter axis. They're also more granular, so they've got more kind of components within them, and as a result sit further up the side scatter. Lymphocytes are obviously the opposite to that kind of morphology, so they sit further down both axes. And then importantly, you also have debris. So debris is going to be little bits of cells that have come about as maybe cells died during preparation or during your harvesting. They're in your sample, but these are going to be smaller obviously in size and obviously smaller in granularity, and as a result they sit down to the bottom left of this forward side scatter axes, and you can gate those out downstream for your when you move on to analyze your antibody information. But I also want to point out as well that while it's really important that we get single cell or single file cells through our cytometer, occasionally that doesn't happen, and that can be for a variety of reasons. But when that doesn't happen, we have to identify that we have doublets within our particular sample. And so we can gate these out using forward scatter area versus forward scatter height. So when you're measuring forward scatter, you kind of get a measure of height and width. You can use those two to measure your area. And then if you plot area against height or area against width, you're able to see where your doublets sit. And your doublets basically sit off of the main kind of population of cells. So most cells are going to pass through as singlets. You can kind of see that nice consistent population here. And these cells that fall off of that main population are going to be your doublets. So we want to exclude those from our downstream analysis. And just to kind of further explain that, you can see an example here where we have two cells passing an interrogation point at one time. And obviously as the first one passes, we start to get an increase in voltage over time. Then that starts to leave the interrogation point. The next one enters and it creates this double peak of voltage over time. And this is how we can identify this particular shift in forward scatter area versus forward scatter height. And then we can gate those particular cells out of our analysis. Okay, so that's the side scatter principles, and then just quickly considering immunofluorescence in the context of flow. So fluorophores encompass a variety of different molecules. These can be either proteins or synthetic molecules. But the important thing is that they accept or become excited by photons of light and emit photons of light in a very predictable manner. So we know exactly what wavelength of light is going to cause this molecule to become excited, and we can expect what wavelength of light that will emit as a result. So we conjugate these fluorophores to target-specific antibodies so we can detect our proteins. And ideally, when we're running our cells through a cytometer, we want to excite them at a maximal excitation intensity, and we want to read that emission at the maximal emission intensity. And this is kind of plotted here. So in this particular example, we're looking at a maximal excitation of around 540 nanometers, but actually anywhere along this particular blue curve, you could cause some level of excitation. But we want to excite this particular fluorophore at the maximal excitation, which is around 540. When we excite this at 540, we're going to hopefully see our maximal emission, which is around 610. And that's ideally where we want our filter to be able to pick up the emission. And the difference between those 2 points is going to be our Stokes shift. Ideally, when you're selecting fluorophores, you want to select those which have as large a Stokes shift as you can find. One, it's beneficial in terms of being able to resolve between excitation and emission. But more so, it's slightly more challenging to find fluorophores with large Stokes shifts. So what it means is when you do find these fluorophores, you can multiplex slightly more easily. So you could include a particular fluorophore with a smaller Stokes shift, and then on the same laser, if you have another fluorophore with a large Stokes shift, you could use that as part of your panel as well. So large Stokes shifts are quite valuable. But what's actually happening inside our cytometer once we've caused this emission of light from our particular cells? So we've got an example here of a detection system set up, LSR 4 TESA, conventional flow cytometer, and it's associated with a 488 laser. On the right, you'll see that we have 3 different types of filters. We have a long pass filter. Long pass filters allow light above a certain wavelength through that filter and onto any band pass filters behind that, which we'll get onto in a sec. Short pass filters kind of do the opposite, so they allow light below a certain wavelength through that filter onto a detector. And then the band pass filters, where you see this kind of 3-digit number and dash 2-digit number, those are going to allow light of a specific wavelength between a range. So a 780-60 bandpass filter will allow light that is 30 nanometers above 780 and 30 nanometers below 780. So a range of 750 to 810 nanometers. Don't be caught out by thinking that 60 nanometers above and 60 below. Always divide that second number by 2, and that will give you your range. So how do we filter out within this detector associated with this one laser? How do we filter out light associated with particular fluorophores into their particular filters and detectors? So imagine we have some light enter here. Oh, I'm hoping that's going to do. Okay, that's not. There you go. Sorry. Okay, so we have light entering, and this light is containing wavelengths of different or a large spectral profile of different wavelengths. As that comes through, it's going to strike the 735 long pass. That's only going to allow wavelength of light above that 735 through onto the bandpass filter. That bandpass is then going to filter further to between 750 and 810, and then that light is going to strike detector A. In this case, it would be PE-Sci7 we'd be reading. The next is going to then be passed along, or the rest of the light is then going to be passed along to the next filter set. The same thing will happen here. Whatever doesn't fit into that range will then be passed onto the next one, and so on until all of that wavelength of light has been read by the various detectors. So effectively, we can have wavelength of light from a large range, and using these filters, we can filter them out so that each detector picks up the emission that was created by a particular fluorophore in your panel. Okay, so onto experimental planning protocols. So in flow cytometry, preparation really is key. I said at the beginning, when you're planning your experiments, this is the area you should spend the most time on, particularly when you're working with larger flow cytometry panels. So we're going to kind of cover 2 areas. One is going to be a little bit about panel design. Panel design is something we could talk for about hours, and I'm conscious of time, so we're going to go through a simple example there, and then we're going to think about our sample preparation and what we need to do when we are preparing to stain our cells with our antibodies. So just a quick note on the difference between indirect and direct staining. So indirect staining is using an unconjugated primary antibody and then a conjugated secondary antibody as your reporter. A lot of people who run flow sometimes think that you don't typically use indirect staining. I would say that that's often the case, but you can use it, and there are use cases for it. So particularly if you're working with a low abundant protein, and that's because using the secondary antibodies, it can help amplify your signal, so it can become a little bit more sensitive for those lower expressed proteins. But there are, of course, limitations. Obviously, doing 2 stain steps means a longer protocol. You need to introduce a new control, a secondary only control, to ensure you're not seeing background from your secondaries. And the most or the biggest limitation when you use indirect labeling is that it does affect your ability to multiplex your flow, because you have to marry up your primary and secondary antibodies based on host species, which can limit your ability to multiplex. And that's why typically most people running flow cytometry like to use direct labeling. And so this is obviously using a primary antibody conjugated to a fluorophore. Obviously, it's a shorter protocol. Just because you have 1 stain step, you often see lower background because you don't have off-target binding from the secondary antibodies. But most importantly is it's way easier to multiplex because you don't have to worry about using antibodies from different host species. But the limitation in any experiment is that sometimes it can be quite challenging to find the right antibody conjugated to the right fluorophore to fit your panel. Okay, in terms of panel design, where do we normally start? So often you'll find that your PI or your supervisor, your boss will say, please look in the antibody box and see what we have so we can start from there. That's understandable, but it's not always the best place to start. And that's because it's really important that we try to find the brightest fluorophore for our lowest expression targets and the dimmest dimmer fluorophores for the more highly expressed targets. And sometimes that's not always what marries up to what you see in your antibody box. We also want to try and space out these fluorophores as best we can over the available filters that we have and the lasers that we have to try and reduce any spillover. So spillover is the case where we see our emission profiles as an example here on the top right, and you can see that there's a little bit of overlap in some cases between these 3 correlite fluorophores in these respective filters. So we're trying to avoid this spillover as best we can. When we talk about brightness, I want to stress that brightness is kind of variable, and it depends on the hardware that you're using, the kind of sample that you're running, and the protocols you're using, whether you're using fixations, the kind of buffers that might be involved. And so just bear that in mind. But typically, obviously, I've highlighted a few examples here which you could consider as a bright fluorophore, as high brightness with PE APC, correlite 647, medium brightness, things like your tandem dyes, correlite 488, and then the lower brightness are things like PerCP and Pacific Blue. But often there's tables that you can find when you're looking at different fluorophores that will compare brightness for you. When you're trying to build panels of antibodies, particularly larger panels, it's really crucial that you understand the spectral profiles of those fluorophores. So the place to start is always going to be using either a spectral viewer or a panel builder. So a spectral viewer is an online tool which allows you to kind of plot the spectral profiles of your fluorophores against each other so you can see where there's potential spillover. But it also is going to allow you to normalize each of those spectra to the lasers one at a time, so you can see how turning on X laser is going to affect your emission profile of those particular fluorophores. Panel builders kind of take that a little step forward. So panel builders always integrate a spectral viewer, but they allow you to kind of build your panel based on your particular cytometer. So you would normally select the cytometer that you're using on the panel builder. And then you can start to even add in exactly what antibodies you already have access to. So that part where I said where you shouldn't start, it allows you to start from that part. You can select the antibodies you have. You can then add in the targets you want to include, and it will recommend what fluorophores kind of fit best within that particular panel. And at Proteintech, we've got both a spectral viewer and a panel builder as well. I'm going to just focus on the spectral viewer today, just in terms of looking at the principles of selecting fluorophores. So I've kind of created a very hypothetical, simple scenario just to demonstrate what it is we're considering when we're deciding which fluorophores to use. So in this particular case, we're using a cytometer which has two lasers, a 488 or blue laser and a 561 yellow laser, and each of those are associated with a particular bandpass filter. So for the 488, we've got 550, 50, and for the 561, we've got 610, 60. And we're looking at two targets, and we have options for three different fluorophores. So we're going to now try and see which of these three fluorophores we should select to take forward into our experiment. So the first thing to do is obviously select the fluorophores on your spectral viewer. In this case, we're using the one Protein has, which is powered by EasyPanel. And it allows you to select the specific fluorophores and view their excitation and emission. The next thing we want to do is start to review exactly where there might be some spillover between these three fluorophores. And we can see hopefully here that we need to be aware of FcZero spilling into PE a little bit. It also spills in a little bit into Alastair 594 in this case. And then P is also spilling into 594. So three potential areas we need to consider. However, as I say, with the spectral viewers, you can actually view what that spectral profile is doing based on the interaction with a particular laser. So in this case, we're showing that we're turning on the blue laser, and we can see here that the blue laser is interacting with two excitation peaks. So it's maximally exciting FcZero, which is fantastic. But there's a kind of double peak with PE in terms of excitation, and one of those falls into the blue laser. So we can see that when we expose these three fluorophores to the 488 laser, that we get an emission of FcZero to a very high degree and PE to a slightly lower degree, albeit within our bandpass filter. So that's going to be spillover we have to be aware of. We can also see that there's minimal excitation of Alastair 594, and that sits outside of our bandpass filter, so it wouldn't be detected in this case. Then we can do the same thing for the 561 laser, and in this case, when I normalize my histogram to 561, I can see that there is no excitation of FcZero, and as a result, no emission. But we do maximally excite PE, and we also excite Alastair 594 to a degree, but both of those are being picked up. So we need to be aware that in 488, we're seeing that our filter is detecting FcZero and PE, but not Alastair 594. And in our 561 situation, we can see that our bandpass filter is detecting PE and Alastair 594, but nothing from FcZero. And hopefully, that will help us identify that in the context of these three fluorophore options, that the best ones to select would be FcZero and Alastair 594, because doing so, neither of those are going to spill over into the other channels. Now, you'll appreciate that we've looked at just three fluorophores in this case, and as your panel grows, this can become an increasingly long-winded and sometimes complicated process. But thank goodness we are living in the advent of AI, which very nicely leads me on to reintroduce Able AI again. I know Frida briefly mentioned Able, but just to kind of reiterate, so Able's our AI system on the website, which you can use to really guide and help you find more easily the antibodies and the reagents that are most beneficial for your particular experiment. So I'm going to walk you through very quickly an example where I used Able to try and help me construct a small panel to help me gate out T cells from PBMC. So in this case, I specified which targets I wanted. I didn't specify the fluorophores that I wanted, and I just said that I was going to be using conventional flow to do this. And within a few seconds, Able very quickly presented me some recommended antibodies that we offer at Proteintech with fluorophores that are compatible in the context of a typical conventional flow cytometer. But beyond that, Able also advised me on some gating strategy considerations. So advised that I should be using some viability dye, which I'll detail in a few slides' time, how I might want to consider singlet gating, so that forward scatter area versus height we talked about earlier, how I might identify lymphocytes from my forward scatter and side scatter, and then some details as to the gating strategy I should employ on each of these four targets, including using CD19, which is a B cell marker, to exclude B cells from my downstream analysis as well. But even more than that, one thing I think is valuable is it highlights some things that we need to consider going into our experiment. So things like making sure you've got compensation controls for these, advising on the kind of titration range you might want to consider when you're staining your cells, and even offering you a staining protocol should you need that to compare to maybe what you were planning to use in the lab. So a really useful tool to not just identify some antibody options, but make sure that you understand why Able has selected these particular antibodies for you. So I think this is really useful, at least when you're starting to build your panels. But I also mentioned earlier that sometimes finding the right antibody conjugated to the right fluorophore can be a real challenge. So Proteintech does have one of the largest product catalogs for fluorescently conjugated antibodies available, so it's a great place to start. But even then, sometimes finding the right antibody conjugated to the right fluorophore is challenging, again, even with Able's help. So what do you do in these cases? I mean, sometimes you might find yourself saying, I wish I could make it myself. And this leads me cheesily on to, well, now you can. So I want to quickly kind of highlight a really, really useful product, which I like in the context of both flow cytometry and immunofluorescence imaging. And that's our flexible antibody labeling kits, which essentially allow you to directly conjugate an unconjugated primary antibody with one of our Correlite dyes. Of which I'll show you the options in a second. But the important thing to note here is that using this kit allows you to conjugate as little as 0.5 micrograms of antibody, which is really quite a small amount, which can be useful for smaller experiments. And you label your antibody in a site-directed manner. So you would be putting a linker on the Fc region of your antibody, which is important because it means the antigen binding domains of that antibody are free to do what we need them to do. And then each of these linkers is also going to carry 5 fluorophores. So it's a known degree of labeling each time. And this protocol is just a 2-step protocol, mixing the linker with an antibody. Then 5 minutes later, adding our quenching reagent, and 5 more minutes later, you're ready to stain. In the context of flow, the real power of using these kits is that it allows you to kind of plug the gaps in your particular panels as and when you need to. So particularly if you already have a panel set up and you don't want to have to reshuffle fluorophores because you can't find the antibody conjugated to the color that you want, using Flexible allows you to conjugate it to the color you need to, and you can put it into your panel in the available laser or filter that you want to use. In terms of the colors, so Correlite is our version basically of the more well-known Alexaflor. We've got 6 different colors. Yes, I had to double check there. 6 different colors, so from 405 to 750, all of which you can use in flow cytometry. We've also got FtZ Plus as well, but I kind of think about that with 488. And I just want to highlight that when you use Correlite, if you're thinking about it in the context of Alexaflor, this is just a quick highlight to anyone who's using spectral flow cytometry, that these are really, really a like-for-like swap in terms of the spectral profiles that you see with Alexaflor, even at a full spectral level. So on the right-hand side is just a table wherever you see 488 versus Alexaflor 488. The closer that number is to 1, the more similar their spectral profiles are. And in most cases, you can see that we're 0.99, 0.98, etc. So these are really, really identical in terms of their spectral profile. And as a result, you could quite easily swap one for the other without having to worry about that too much. And then just very, very quickly, I know we've got an immunofluorescence talk tomorrow, which Frida will be leading, so please join for that if you can. But for those of you who might not be able to join, I just want to stress that you can also use Flexible in the context of multiplexing your immunofluorescence staining, in that you can primary conjugate your antibodies regardless of, or you can conjugate your primary antibodies and then downstream regardless of their host species. You can use them together. So if you had, for example, these are 5 rabbit IgGs as an example, you could label each of these with a different color. Once you've labeled these, you can mix them together and stain in one stain step without worrying about having to use secondary antibodies to prevent that cross-reactivity. So I just wanted to highlight that for anyone who's doing immunofluorescence as well. But back to flow. So that's kind of shooting through some kind of tips for panel design. Just to reiterate, we always want to choose the brightest option available to us, but when you're working with large panels, you do have to kind of compromise. So where you are using antibodies against lower expressing proteins. You want those conjugated to the brightest fluorophores. And the reverse, obviously, for when you're looking at lower expressed proteins, you want those, higher expressed proteins, you want those with dimmer fluorophores. A couple of other tips as well is if you're working with intracellular targets, sometimes it's good to use brighter fluorophores for those, just because they can be slightly more difficult to stain than extracellular targets. If you're working with proteins or cell populations which can be clearly distinguished, i.e. they form two kind of different populations on your dot plots, then sometimes you can get away with slightly dimmer fluorophores then as well. If you're using tandem dyes, so things like PE-Cy7 or APC 5.5, when it comes to intracellular targets, these dyes have slightly larger scaffolds, and so sometimes it can be a little bit harder to penetrate through the cell, particularly the nuclear membrane. So if you can avoid using those for those particular targets, then I'd recommend doing that as well. It's not a super strict rule. It's just where possible. I would suggest that, and just be aware if you're seeing any background, it might be the result of struggling to wash away those tandem dyes after staining. But the big one really is ensure that you're using a flow cytometry panel builder or spectral viewer in order to support you in your panel design. Okay, so we've done a lot of preparation, a lot of planning, and now we're moving on to the wet lab stuff. So we're at the bench and we want to start getting ready to stain our cells. So the first thing to highlight is it's really important that we try and preserve our cells in as natural a state as we can, and we want to maintain as high of a viability as we can. So basically, we want to keep our cells alive until the point that we need to fix them, if we're going to fix them. So ideally, we want to process cells as soon as we've collected them, and we want to tailor our dissociation steps if we're working with tissues based on the cells that we're trying to isolate. So sometimes we need to consider a slightly more harsh dissociation if we're using a particular tissue, or we can get away with using a slightly more gentle dissociation if we're using another type of tissue. And that really depends on the biology of your system. So just ensure that you're doing your kind of literature searches based on that. We also want to ensure that our cells are in a single cell suspension, and the way we achieve that is using cell strainers to avoid clumping of cells. So using between 70 and 100 micrometer cell strainers will help you have a single cell suspension rather than clumps. Keep your cells cold. Prior to fixation, the best way to keep them alive is keep them cold and keep them in the presence of FBS or BSA. So your FACS buffer will be 2 to 3% FBS or BSA made up in PBS. And really important is always perform a cell count prior to staining, just to understand how many dead cells you might have in your sample. Dead cells really, really are the enemy. And ideally, we want to try and work with a sample which has got about 80% plus viability. 80 to 90% is typically what we want to aim for if we can. In terms of immunostaining, it's really important to try and understand the biology of the proteins that you want to investigate in the context of where you expect to find them within the cell. I know often people are kind of handed a protocol by postdocs or a PI or wherever it may be, and you're kind of just told to follow that. But sometimes, if you're adding new targets into a panel which was designed for extracellular staining, then you need to consider if you need to introduce any fixation and permeabilization to access those particular targets. So there are kind of differences in the protocol steps that you would follow. So extracellular target staining will always obviously start with your cell isolation or dissociation. Then you would run a viability dye stain. If you're going to keep your cells alive, you can also do this towards the end of your protocol too. Then you'd follow this with a serum or FC block to try and prevent any non-specific binding of your antibody to your cell. Then you would go into your antibody stain, your antibody panel stain. You do have the option of doing a fix if you want to do it during your extracellular staining before you go into analysis. If you are fixing your samples, I would strongly recommend that you don't keep them for more than kind of 7 days within the fridge. After that time, I think sometimes you need to consider whether or not your antibodies are going to perform as well as they would have done, the closer to your stain day. In terms of intracellular target staining, what we need to consider here is that we want to stain our extracellular targets first. So we follow the protocol up to that point, but then we want to fix our cells first, and then once we fix them, we want to permeabilize them so we can access the intracellular targets there. And the selection of fixation and permeabilization depends again on whether or not you've used extracellular antibodies. So if you have stained for extracellular targets, you might not want to use something like methanol, for example. You'd want to use something like PFA. And then when it comes to permeabilization, if you are staining targets which might be cytoplasmic, you could use a slightly less harsh permeabilization, something maybe like saponin, or a little more harsh like 0.1% tritin. But if you're looking at intranuclear targets, often it's better to use buffers and permeabilizing reagents designed to access the nuclear membrane. And usually those are supplied as commercial kits optimized for that very purpose. And that's definitely something that Proteintech have on our website as well. So please do check that out if you are working with intranuclear targets. Just a couple of quick tips. One thing I can't stress enough, and this is something I learned after my PhD as I moved into industry, was antibody titration really is important, and it does affect the quality of the data downstream. So when you're introducing new antibodies to your panel, or you're starting using a new product, make sure that you titrate it to find the right concentration to use for your particular sample. I've kind of mentioned keeping your cells cold and the viability you want. Blocking your samples, use either FC block or serum. I'm going to go into some more detail on that in a second. In terms of how many cells typically are stained in a flow experiment per sample, it's usually around 1 million, and that will be in a volume of around 100 microliters of stained solution. And I've obviously commented already about how long you can keep your cells after fixation. It just allows you a bit of flexibility if you've had a long protocol and you can't get onto the cytometer that same day. Okay, viability dyes. So again, stressing it's really important that we try and keep our cells happy and healthy, and viability dyes are going to tell us just how well we've managed to achieve that. So viability dyes are used to identify if a cell has died or not. Dead cells are a real problem because they can lead to non-specific binding of your antibodies, which is obviously going to cause some background signal. Dead and apoptotic cells are also autofluorescent, and as a result of DNA release can cause cells to clump, which is a nightmare for your flow cytometer. And so we use these viability dyes to see just how well we managed to do it during our cell prep, and ensure that we look at cells that were live to know that the signal we're looking at is truly specific. So just two things to highlight here, and this is really in the context of whether you're running your cells live or whether you are going to fix your cells. So if you're going to run your cells live, you can use DNA binding dyes. So things like DAPI or propylium iodide are options for those. But if you want to fix your cells, then you need to use a viability dye compatible with fixation. And these would include things like phantom dyes from Proteintech. Effectively, the difference here is between the two is that with DNA binding dyes, obviously, if your cell is alive, the cell membrane is intact, so they don't bind to your nucleus and you don't see a signal. But if that membrane degrades because your cells died, those DNA binding dyes access the DNA in the nucleus, and as a result, you generate a signal. Phantom dyes are slightly different in that they are amine reactive. So they bind to amine groups present on proteins, both on the surface of your cell and also on the inside of your cell, but they can only access the intracellular proteins if your cell membrane is not intact. So you'll always see a signal from phantom dyes when you're staining your cells, but a large shift will identify that those cells are dead compared to the ones which are alive, because there's more dyes binding to protein in that case. So here's an example of gating out your alive cells, which are slightly more negative for fluorescence compared to your dead cells, which sit a little higher up that fluorescence axis. One thing as well to highlight is when you're running phantom dyes, because they're amine reactive, you don't want them to be, you don't want to stain your cells in the presence of BSA or FBS. So at that point, I normally recommend doing a PBS wash prior to your stain, then making your phantom dye up in PBS only, staining your sample, and then once you've done your wash, you can reintroduce your FAX buffer containing BSA and FBS for your blocking step and your subsequent antibody staining downstream. So we've mentioned blocking a fair amount. In flow cytometry in particular, there's a blocking need for Fc receptor binding. So particularly if you're working with immune cell populations, if you're working with PBMCs, if you're working with tumor samples, anything that you might find immune cells in, it's really important that you consider whether or not you need to use Fc block. So things like monocytes, macrophages, granulocytes, B cells as well, NK cells and dendritic cells all express Fc receptors. And on the right, just to kind of very quickly detail this a bit more. So this is how we want our antibody to bind specifically to our target protein. These Fc receptors bind to the Fc fragment of your primary antibody, which would lead to a non-specific background. So effectively what we do with Fc block is we introduce this kind of Fc fragment effectively, and what that does prior to your antibody staining is it occupies those Fc receptors so that they can't bind non-specifically to your primary antibodies, and as a result you get a nice specific signal as a result of that. So this is kind of what you would expect to see. So in this particular case we're looking at PBMCs. So we have CD4 on the y-axis, CD3 on the x-axis. This particular population of cells is monocytes. Monocytes do express CD4, but they don't express CD3. So what we're seeing here in the absence of that Fc block is a shift in the population of monocytes, which is non-specific. But then using Fc block, you can see that that shift is dramatically reduced, so that we have far less background in our gate, although arguably there is still some background, and that is something that Proteintech have very cleverly worked out how to fix. That is this is a product I'm pretty excited to present to the flow community. So FcZero is our group of recombinant antibodies which we've designed specifically for use in flow cytometry, and the clever thing that our scientists have done is mutated the part of the Fc backbone of our antibody that Fc receptors usually bind to, so that those receptors can no longer recognize the antibody. So it eliminates Fc receptor binding. We also use an AccuBright system, which effectively is a site-specific conjugation. So we know where our dyes are going when we label these antibodies, again, protecting your antibody binding or your antigen binding sites, so we know the antibody can still what it needs to do. And just to demonstrate how effective these are, so we've recognized these two dot plots from previous slide. You can see here that when we introduce FcZero, even in the absence of Fc block, that there's a complete removal of any of this non-specific background. So I would highly recommend, if you're working with cell types that express Fc receptors and you can use FcZero antibodies, that you consider adding these into your panel. And I just want to stress as well very quickly that within the flow community, often we have preferred antibody clones that we want to use. So a particular CD3 clone or a particular CD4 clone, for example. And so what we've done at Proteintech is ensure that we can build those clones into our FcZero portfolio, so that we retain the same epitope binding behavior of the clones that we know many flow cytometry scientists like to use. So if there's a particular clone you're interested in, you can have a look on the website to see if it's in the FcZero portfolio, and if it's not, let us know, and that might be something we can build into it in the future as well. Great, so we're making good progress through our agenda, moving on to flow cytometry controls. Now, this really is an area I can't stress enough. We want to spend some time considering and making sure that we're building in the right controls into our experiment. I'd argue that in many experiments, particularly again if you're using large antibody panels, that this might be the most important aspect of a multiplexed experiment. So often without these, you might struggle downstream to differentiate between true positives and potential false positives or background. So your controls really should never be an afterthought. And I would argue that in some cases, they're almost more important than your test samples, because without them, you can't get the data you need from your actual samples. So there's an array of different controls you might want to consider. I'm going to go through 4 ones which I believe to be key, but I'm confident that there's probably a few more that you might want to consider depending on how your protocol or your study changes. But we're going to start first of all with compensation controls. So compensation controls are incredibly important when you have several different fluorophores in your panel. And the idea with compensation controls is often, we spoke about spillover earlier on, and spillover is something that we try our best to avoid, but often we can't avoid it completely. And so using compensation controls will allow us to correct for that potential spillover. So when you're running compensation controls, it will involve you using one antibody from your panel to stain your sample, and you do that for each antibody within your panel. So if you have a panel of 10 antibodies, you're going to have 10 compensation controls and an unstained control as well. So you do that for each antibody. One important thing to note is make sure you stain enough cells. So I would always say try not to let this be done at the end of you aliquoting your cells into your different wells. Try and make sure that you've got enough cells to stain for your compensation controls. You always want your signal to be as bright or brighter in your compensation controls than your samples. So ensuring you have enough cells to stain is going to help with that as well. Okay, so I just want to highlight an example here. Again, we looked at FcZero and PE earlier, and then I kind of implied then that those couldn't be used together, but the reality is that using compensation means that they can. So if we have a look at this particular dot plot here, we can see that when we plot FcZero against PE, so in this particular sample, there is no PE present in the stain, there's only FcZero, and this is before compensation. So we can see that because FcZero does kind of spill over into the PE filter, that we get this kind of weird 45-degree tail. And this tail is really indicative that there is compensation required and that there is spillover. So if you're running experiments and you kind of see these sort of tails, then that's a good indication that there might be compensation required, or that you need to review your compensation controls. And by running our compensation controls and using the formula that your analysis software will integrate into its system, you will be able to correct for that spillover, and ultimately it will it will manage the spillover so that it corrects it back to where it should be for just FcZero. So it basically corrects for this spillover that we see in this particular PE emission filter. And so this is what we would be basically doing for each fluorophore within our panel. So if you have 10 fluorophores, you'll be doing this for each of those one at a time. Some tips for compensation. As I say, it's very important, so almost more important than your samples. You want to make sure you collect enough events or enough cells. But the key here is that you see clear peaks and clear separation of your peaks between a positive and a negative population. Your control signal should always be, your compensation control signal should always be as bright or brighter than your samples, but never weaker. Ideally, you always want samples that include positive and negative peaks. So that might be intrinsic within your particular sample. So if you're using PBMCs, for example, you'll have CD3 positive cells and CD3 negative cells, so that would be fine. Where that's not the case, sometimes you can get away with using an unstained control as a universal negative control. You can also make the decision to use compensation beads, where you will have a positive bead that does bind to your antibody and a negative bead which doesn't, and those will form two separate peaks when you are looking at your at setting your compensation gates on your hardware. So one thing to make a note is, where possible, use cells, but for targets that have low expressing proteins and you need to generate enough signal to compensate, then using compensation beads can be a good alternative to cells in those instances. But where possible, try to use cells as best you can. Ideally, you do want to compensate. You do want to run compensation controls fresh for each experiment, and that's just because of drift that can happen in the performance of your machine over time. So if you're using compensation controls, even if it's on the same panel from months ago, and you've maybe got a new lot of a particular antibody, that can affect the compensation of your panel. And it's really important that when you are running these controls, that you use the exact same fluorophore as the one that you're using in your test sample. So FcZero is not suitable as an alternative to Coralite 488, for example. The other thing to note is if you're using tandem dyes, then the compensation control should be from the very same lot of even the same vial. And that's just because those particular dyes can sometimes over time degrade, and so you want to ensure that you're using it from the same vial to make sure that those compensation values are as accurate as can be. So the next control we'll introduce is fluorescence -1, or FMO controls. The way I think about these is they're kind of the opposite to a compensation control, in that rather than having one antibody stain per sample, you're now actually staining all of the antibodies in your panel on your sample minus one of them, and you're going to do that for each target within your panel. So the idea for these is it's going to help you set your gates downstream, so making sure that what you're looking at is a true positive and not any kind of background or drift just from the performance of the cytometer. So as an example here, if I look at the CD3 FMO, we're going to have everything in this particular panel, for T-cell panel, except for CD3. For CD4, we remove CD4, CD8, we remove CD8, etc. And then in the context of what that kind of does for us, if we look at this example for FOXP3, which is a Treg marker, we can see that for CD4, we have a nice clear shift, as we would expect to, because CD4 is present within our FMO, but we've excluded FOXP3. So we would hope that over here, we would not see any cells, which we for all intents and purposes, don't. But then when we introduce FOXP3 to our full panel, then we do find that we've got a nice shift in CD4 FOXP3 positive cells, denoting T regulatory cells. So this is just highlighting that we can trust that this signal is true. And it's particularly important if you're going to have rare events, because the rarer an event, the harder it is to work out if it's a true positive or potentially false positive signal. So FMOs are incredibly important, particularly for multiplex panels as well. Another type of control is an isotype control. I know a lot of people are hesitant of these. I think they're really useful as long as you're not using them to gate populations. You should use FMO controls to gate your particular populations. But in terms of validating your antibody and making sure that what you're seeing is a true positive signal and not the result of non-specific interactions of your cell with your antibody, isotype controls are a great tool to use. So they help distinguish your positive signal from potential background or non-specific interactions of your cell with your antibody. They're really useful for when you're titrating your antibody to ensure that you are looking at the best concentration with the optimal separation and minimal amount of background. So really strongly advise using these when you're titrating your antibody. Importantly, these should always be the same clonality, the same isotype, and conjugated to the very same fluorophore as your target specific antibody. You should always also use these at the same concentration, because what you're trying to do is just see how your cell is interacting with an isotype control antibody or with an antibody in a non-specific way. And these isotype controls are designed not to bind anything in your sample. And so any signal that you see generated is the result of non-specific background caused by your cells interacting with the antibodies that you're putting onto them. So as an example here, we've got an unstained sample. Then we've introduced an isotype control. You can see that there is some change in the profile. So there is some kind of interaction happening of our cells with this particular isotype control. But then when we compare that to the target specific antibody, so in this case CD4, we see that there's a very clear shift in monocytes here and CD4 positive T cells here. And when we plot that as histograms relative to our isotype control, we can see that there's clear differences, and that allows us to confirm that what we're looking at is true, is a true positive signal effectively. And then finally, just on controls, a very simple one here is just the idea that if you're using treated cells, it's important that you include an unstimulated control as well, just to make sure that what you're looking at is the result of the treatment and nothing else. So just ensuring that you subject those cells to the same staining process as your treated cells as well. Okay, we're very nearly there. For those of you who have managed to hold off until the end, we're going to do some real quick troubleshooting. I am conscious of time, so I'm just going to go through these, not all of them per table, but we can definitely touch on a few of these in the Q&A as well. So just to begin with, what happens when you have weak or no signal in your results? So the first thing might be that your antibody concentration just was maybe too low. So I would again urge you to titrate your antibodies prior to your main experiment and just see what works best to ensure you get the best and brightest signal. Sometimes if you're running treatments, it can be that your treatment didn't induce the production of your target protein. So you might want to review treatment conditions for that. Sometimes you can also introduce a positive control antibody in that scenario. So an antibody against another target that you know should increase as a result of a treatment, but that's different to your actual target. And that way, if you see binding to that particular target, you know that there might be an issue with the antibody rather than your treatment. Weak fluorophore with low expressed targets, so always try and use the brightest fluorophore you can. As I say, again, there is some compromise you have to make, all dependent on the expression of your protein. So if you've got a low expressed target, make sure that you're using a bright fluorophore for that particular example. If we go through some quick troubleshooting for some high non-specific and background signal, so similarly to low antibody concentration, always titrate your antibodies. So if you have really high non-specific signal, try and titrate those antibodies to the optimal point. That might reduce the non-specific signal you're seeing. We mentioned dead cells can cause background as well, so make sure you've included a viability diet. Make sure you've also done your cell count prior to staining to ensure that you've checked that there aren't too many dead cells in your sample. For autofluorescence, this isn't something we touched on in the talk, but one kind of pro tip is if you're using autofluorescent cells and you want to look at lower expressed targets, sometimes it is beneficial to put those in a far red channel rather than a green channel, just because the autofluorescence can kind of block out that signal in the 488 channel. So that's one thing I would say is consider using the far red channel if you can. Inadequate washing is definitely something as well that can cause background, particularly when you're doing intracellular staining. So ensure that you're doing adequate washing. And if you need to include detergent in your wash, then you can consider doing that as well. And then finally, we touched very quickly on compensation. So one thing to look out for in terms of poor compensation, is checking if there's any kind of 45-degree angle tails in your plots, and if there are, then you might want to just review your compensation controls and consider the best course of action based on what that data is telling you. And then just to very quickly summarize, this is not summarizing the whole talk, but just more my kind of thoughts on flow. So flow cytometry really is a really powerful assay that will help you to analyze many different cells from a particular sample. So it's highly multiplexable, it's semi-quantitative, and it's definitely robust. So it's a really useful tool to use. Controls is the thing I want to stress most of all, is make sure you consider the controls that you need to use to kind of compensate and also to gate on your populations. And then I think it's important to say that it is a complicated technique, particularly when you're first introduced to it. So importantly is don't be afraid to ask any support in your institutes or your places of work that are using flow cytometry already, and just ask them for guidance, and hopefully they'll be able to help you in the first kind of exposure to working with flow, and make sure that you're able to get the most out of your data. And with that, I am all finished. So a big thank you for listening. I think we'll hopefully have a look at some questions. I think, Afrida, have you got a few slides, did you say?
Yeah, just a few more. First, thank you so much for that presentation. I learned a lot. It was a very great comprehensive review. You can check out the comments. People are very appreciative of all your efforts. So definitely take a look at those comments. I think someone said that they struck gold, which resonated with me. But yes, I have just a few more slides before we get to the Q&A. I will make them quick because I know we're a little over. All right. So before we jump into the Q&A, I really just quickly want to highlight a few more products that kind of complement our flexible and FcZero products that Ali mentioned in his talk. So I'll begin with just kind of an overview of our recombinant flow antibodies. So in addition to polyclonal and monoclonal antibodies, Proteintech also offers a very wide selection of recombinant antibodies that are validated in and also optimized for flow cytometry. So all our recombinant antibody clones are carefully selected after going through multiple rounds of validation in order to identify clones that have the best signal intensity and that offer the lowest background. So this includes antibodies from top-sided clones that are produced with rabbit and mouse host isotypes. So you get the epitope and the binding affinity of that gold standard clone that you know and love, but on the recombinant backbone that's going to fit your panel. Our R&D team has also genetically engineered new recombinant antibody clones that have optimal performance in flow. And then, of course, as Ali mentioned, our FcZero antibodies, which have the mutated Fc receptor binding domain to eliminate that need for Fc receptor block. And then finally, I just want to mention that we also offer nanobodies for flow cytometry for immuno-oncology targets. And since these products are all recombinantly manufactured and rely on cloning into high yield expression vectors, you could get all the benefits that come with recombinants. So you get the consistent performance, the high lot-to-lot consistency, and the unlimited supply. So all our antibodies for flow are conjugated to either conventional dyes, or we also have our in-house Coralite Plus dyes, which are bright fluorescent dyes that offer high photostability and minimal fluorescent bleed through. And what this allows you to do is use several of these dyes in a single panel. So these dyes are going to be equivalent in spectral profile to Alexa Fluor dyes and undergo stringent validation to ensure reliable results. And then this chart here just shows this chart here just shows an overview of the dyes that we offer, and then also their laser compatibility. So as I mentioned, we have our in-house coralites as well as antibodies that are conjugated to conventional dyes like FITC, PE, APC, and other dyes such as Atlantic blue, which is going to be spectrally similar to Pacific blue, or then we also have cardinal red, which is spectrally similar to Texas red. If you're starting out with your panel design, Proteintech also offers pre-optimized flow cytometry panels. So these panels, which are listed here on the bottom left, these panels were curated and optimized by our R&D scientists to detect specific subpopulations of cells. So each panel incorporates top-sided clones and is designed to have that PE channel open for you so that you can easily incorporate your target of interest into that panel. And then finally, I just want to mention that Proteintech also offers supporting resources, reagents, and kits for flow, including isotype controls, viability dyes, cell health assays, and various buffers. We also offer a wide variety of blogs that cover topics from panel design, gating, and data analysis, as well as protocol guides and past webinars from internal and external speakers. We also have resources to help with panel design, as Ali mentioned. So we have our Spectra Viewer to help with multicolor panel experiments, as well as our panel builder, where you can set your specific cytometer and enter panel details for a customized panel design. So if you found this webinar useful and you want to see more, please do check out the video hub on our website as we post all our past webinars there. We also have an online learning portal, which offers free expert-led training courses to researchers. So currently we have topics covered like image analysis using ImageJ, statistics, Western blotting, and R coding. And we have many other courses coming soon. And then lastly, don't forget that today was just day one of Immunoassay Applications Week. So we also have more application workshops coming up. So please do sign up for the other ones if you found this webinar helpful. And then as an attendee of today's webinar, you'll receive a couple follow-up emails. The first one will have a certificate of attendance for this webinar, and then you'll receive another email afterwards after the Immunoassay Application Week has ended, and it will have a recording of all the webinars as well as a exclusive promo code for everyone who's signed up for our Immunoassay Week. It is 25% off one full-size antibody using this promo code right here. All right, so with that, I think we can go ahead and start the Q&A. If you do have any other questions that we don't get to, you can always email Alastair directly. I included his email right here, or you can always reach out to us at events@ptglab.com. And please do follow us on social media for all our upcoming webinars because we post about everything on our socials. So with that, I have a few questions that I think might be good to address. So we had one question coming in that asked, what strategies help reduce nonspecific binding of secondary antibodies during indirect staining?
Great. That's a great question. So the first thing I would say is there is blocking when you're using secondary antibodies. So usually on your secondary, you will have it as a something anti-something. So for example, you might have a goat anti-mouse. The species in which that secondary was raised, in this case, is goat, and so the ideal option to use, I think, is to use the goat serum in that particular example. So I would just make sure that you marry up the serum that you want to use with so you use the serum block with the host species that secondary was raised in, and that will help in terms of your blocking. The other thing, of course, is again antibody titration. So titrating your antibody as well will try and reduce any non-specific signal. It will reduce non-specific binding because it's less antibody. So make sure that you're titrating and make sure that you're blocking your cells as well. And that blocking step again will happen during the prior to primary antibody staining as well.
Great, thanks. The next question is asking whether or not cells get damaged while passing through the laser. Is this a consideration for sorted cells for further culturing?
Good question. So in terms of the laser causing damage to the cell, not typically, no. The laser shouldn't cause damage to the cell. But one thing you do want to consider is one, different cells are more or less robust than others. So cell sorting processes can be really useful for some cells and not for others. But the other thing to think about is the flow rate to which you subject those cells to. So usually on most bits of hardware, you'll have a low, a medium, and a high flow rate. The higher the flow rate, the increased kind of amount of pressure within the core stream, and ultimately that can potentially cause damage to your cells as a result of that. So where possible, you want to reduce the flow rates as well.
Great,
but the laser itself shouldn't cause any particular damage, no.
Our next question is asking if phantom dyes react with both dead and live cells, then how are the live cells separated from the dead during analysis?
Yes. Okay. So this is maybe I didn't explain this as well. So phantom dyes and fixable viability dyes in general react by binding to amine groups that are present in proteins in general. So when your cell membrane is intact, those phantom dyes can only bind to the proteins on the surface of your cell. They can't access anything inside your cell. And so you get a reduced fluorescence compared to when you stain dead cells, where you have phantom dyes staining both the extracellular proteins, but also binding to amine groups on the inside of the cell. So you have an increased fluorescence. And so you do get fluorescence in both cases, but for dead cells, that fluorescence is far higher. And as a result, we see a big shift in that cell population relative to live cells, and we can gate out the dead cells as a result of that.
Great. If I already have an isotype control and a stained sample, do I still need an unstained control? Is there a preferred acquisition order, for example, stained before isotype control?
I would always run unstained controls with every experiment. So I would use unstained, I would use an isotype control, and then I would use your stained sample, your normal target antibody sample. The isotype controls usually are when you're introducing a new antibody to an existing panel, or you're only using one antibody within your stain, and you want to ensure that you're looking at specific signal. I would always include the unstained control, because when you plot the isotype over the unstained as a histogram, you'll be able to see if there's any significant shift. And what that will help you do is downstream, as you titrate your antibody, you'd hope that your isotype control will drop down the axes as you reduce the concentration so that it overlaps with your unstained control as close as you can get it. And hopefully at that point, you'll still see a nice significant shift in your actual target specific antibody as well. And that would be the point that I would say, okay, this is likely to be a suitable titration or a suitable concentration to use this antibody at. I should stress though, if your isotype control does shift a little bit off of your unstained control, but there's still a clear shift in your actual target antibody, then that's still compatible. You don't need to have no shift in your isotype. You just need to make sure that your isotype and your antibody against your target don't overlap. That's the key.
Great. I think we have time for maybe a few more questions. So this one seems like a two-parter, so I'll start with the first part. So they were wondering if you had any experience on working with nanobodies conjugated with fluorophores for flow. So what are the differences when working with conventional antibodies?
Great question. So I think you're speaking to the right company. We're really excited. Obviously, Frida mentioned that we have nanobody products produced at Proteintech, and increasingly we're trying to drive the range that we have acting as primary nanobodies. And many of these we have as immuno-oncology targets, and we are conjugating those to fluorophores for use in things like flow cytometry. I would say maybe in terms of benefit relative to antibodies is because these are smaller, they are a little bit or they find it a bit more easy to access your cells in particular. So you have less of this problem with larger scaffolds of fluorophores because the nanobodies are 10 times smaller than an antibody. So that can be a benefit. But more so than that is when you are washing cells after intracellular staining, is once they've bound to their target, if there's any nanobodies which haven't bound to the target, they're going to be able to wash out more easily. So that should reduce some background as well. So yeah, that's what I would say in the context of nanobodies is if you have access to them against your particular target, then that's fantastic. And if you're working with immuno-oncological targets and a few cytokines, then Proteintech certainly have some options there for you as well in flow cytometry.
Great.
I don't know if you want to add anything to that, Frida. I think that's...
No, I think
that
was great. And then the second question is how can I prevent unspecific binding once I've tried different buffer conditions, viability dyes, etc., for tumoral cell staining?
Okay. Tough question to answer without knowing a bit more detail on the experiment protocols and the specific cells that you're using. One thing I would say is if you're using tumor cells, they are notoriously difficult to work with in the context of flow, as I experienced. Importantly, I would say, and I've kind of said this already, is one, ensure you're blocking your samples correctly. So in tumor tissues, you're going to have a lot of immune cells. Make sure you're using FC block and make sure you're using it at the right concentration. So in a way, you can kind of titrate FC block as well. You can increase or decrease the concentration, apply the same concentration of your primary antibody and see how that background changes, similar to the dot plots that I presented for the FC block earlier on. And the other thing I would say again is down to antibody titration. Again, if you're seeing background, often that's the result of not titrating the antibody correctly. If you try to titrate the antibody and you still see there's a problem, then it might be that you want to consider sourcing a new antibody. Ensure you're using a recombinant or a monoclonal antibody rather than a poly in that particular instance, just to ensure that the specificity is against that one epitope to reduce any non-specific binding. So those would be my kind of tips, but I think I need a little bit more information to kind of guide a bit further.
Great. So let's do one more question. So the last question here is asking about freezing PBMCs. So what are your thoughts on freezing PBMCs, defrosting, and then staining for flow? Do you have any tips?
Yeah, so I think again, this is one that kind of depends on what you're doing with them downstream. If you are going to try and culture them, you can do it, but I would stress that you want to track the viability of those cells. So you might want to see, if you know you're going to have a protocol which is going to last a week, is track the viability of those cells over the course of a week to see if you're going to be able to keep them alive for that period of time. Freezing PBMCs does often lead to poorer viability, not just PBMCs, but many primary cells, in my experience anyway. So ideally, you would culture them from fresh, but I appreciate that can add in a bit more time into your day. But really, it depends on what you want to do downstream. If you're not going to culture them after you've frozen them, you're just going to, thaw them and run a particular panel on them, then that might be okay. Again, just make sure that the viability post-thaw is suitable. So you ideally are going to look for around 80% viability.
Awesome. Well, great. Thank you so much. I know we have so many questions coming in, and I apologize that we cannot get to all of them live. I'm just going to share that last screen one more time so that you guys all have Alastair's email in case you want to email him directly. I'm sure he would not mind if you guys reached out with any questions that you have. So with that, I will go ahead and end the webinar. Ali, once again, thank you so much for that informative talk. And thank you everyone who attended. And don't forget that we have workshops every day this week. So make sure you take advantage. So with that, I will go ahead and end the webinar. And I hope everyone has a good morning, afternoon, or evening, depending on where you're tuning in from the world. Take care.
Thanks.